So we take leaf samples, we send them off to the lab presses those leaf samples and extracts the sap from inside those leaves, and they're analyzing the nutrient content of that. There are some specific protocols to sampling. We are sampling old leaves and young leaves separately once we've got enough growth to do that. And it is quite a bit different than a conventional tissue test. Here are some of those differences. When we're looking at a tissue test, I like to say that we're looking a little bit backwards. And the reason I say that is when you look at a conventional tissue test, a significant portion of that test, roughly around 80% of it, is actually what we call leaf dry matter, leaf tissue. That leaf tissue has nutrient inside of it that's being analyzed because we're analyzing the whole leaf, right? But that leaf tissue that has the nutrient, that nutrient is no longer available for that tree growth and for that crop development. It's essentially utilized, it's tied up. It's the sap that actually has the nutrient that's available for the crop. A tissue test typically has about 12 to 13 different data points. And when we go out into the field and we sample for a tissue test, we're typically doing it in and roughly around the middle of the new growth. And that's going to give us a bit of general data. And the reason for that is nutrients move in the tree. Some nutrients move very little, some move a great deal. And we want to be able to capture that. So with the sap analysis, I like to say we're looking forward. And the reason for that is we now get rid of that 80% leaf tissue that has nutrient, but that nutrient is no longer available. We're zooming in, we're just focusing right in on the nutrient that's in the leaf sap that's still available for that crop and for that crop development. Typically, we've got 23 different data points. And because we are sampling old leaf and young leaf separately now, we're seeing nutrient mobility. We're seeing what's happening as the nutrients are moving in the tree, and we're catching those imbalances before they're actually visible. The lab we're working with is NovaCrop. One of the key reasons why I like NovaCrop, because there are now a few labs doing sap analysis here in North America. NovaCrop is in the Netherlands. NovaCrop seems to be the only lab right now that has specific targets. A lot of labs are doing sap analysis now and comparing the old leaf to the young leaf, basically called comparing leaf differentials, which is somewhat good. But what if the old and the young leaf are both really high or really low? You're going to miss that. NovaCrop actually has specific targets for cherries, for pears, and for apples, among a number of other crops. So not only now can we compare old leaf and young leaf, we're now comparing towards a standard set of targets on where we want those actual numbers to be in the leaf. So sap analysis has given us new knowledge into what's actually happening inside the tree. We're getting it timely in that it's real-time data to what needs to be done now. If we follow the protocols that have been outlined on how to take a sample, we're getting accuracy. And now we're also getting precision. So accuracy is that we're getting solid results. Precision is we're able to do that over and over again. Think of accuracy and precision as accuracy is if I can throw the dart at the dartboard and get to the center. Precision is if I can do that over and over and over again. And we're now able to do that on sap analysis. And that's given us efficiency. We're wasting less fertilizer and we're maximizing crop yields. Apple grower, forgive me, I know this is a cherry group, but a good example is I'm working with an apple grower out east, and a fairly large apple grower. He's got about a $500,000 fertilizer budget. So fairly substantial budget. Through utilizing sap analysis and combining it with his fruit analysis and combining it with his soil analysis, we discovered that potassium was a real hindrance for him. He was getting lots of K coming out of the ground, and it was actually something he didn't need a lot of, yet it was a significant portion in his program. By removing that from the program, his crop went up in quality, yield stayed consistent, and he saved about 100 grand a year in his fertility bill. So improving fertility doesn't necessarily mean we're going to add more fertilizer and spend more money. It can also mean the opposite. As a matter of fact, in my time doing SAP and working with growers across the country, I'm finding exactly that, is it's often the imbalance is being created due to an excess, and we need to take something out of the equation, not add something. I'm not going to go through too much with NovaCrop crop. If you're interested, let me know. Let's get into the Sap Analysis Report, because this is where we can talk a little bit. So when we look at a Sap Analysis Report, now this is one from Nova Crop, and I've got it broken down a little bit into the order in which they've got it on the reports. When you look at this, the light green bar is the young leaf result. The dark green bar is the old leaf result. The target is to be somewhere in the optimum range, right in the middle. To the left would be somewhat deficient. To the right of that would be getting into the realm of toxicity. Now, it starts off, we look at sugars, pH, and EC. Now, this is in the sap, right? So we're looking at the sugars in the sap. So that's giving us an idea of how the leaves are functioning. The pH is giving us an idea how those main cations are in the sap. So we want to be thinking about magnesium, potassium, calcium, and those ratios that will reflect on the pH. The EC gives us an idea of the electrical conductivity. It's the same as in the soil. If there's a lot of mineral, a lot of nutrition hanging around in that tree, we're going to see it in the EC. So is there too much or is there not enough? And then we'd go further down into the sample to see where that excessive or deficiency might be occurring. Potassium, calcium, magnesium, sodium, and ammonium are also looked at. So this is where your potassium-calcium ratios come into play. We really want to watch that early season potassium. We don't want that high. We want to be building on the calcium. Magnesium. Magnesium is something, by the way, I've seen it now 2 years in a row. Early season magnesium has been deficient, everyone. Quite deficient. And that speaks to post-harvest, because with cherries, we've got bloom happening really before those flowers are out, and that early leaf development, once you're well into bloom, is being driven by what you did last fall. So when we say the first fertilizer application of this year is your post-harvest last fall, this is what we mean by that. And I'm going out early season and I'm already seeing a lot of magnesium deficiency, and we can help prevent that by putting a good foliar magnesium in your post-harvest last year. Realizing that post-harvest sprays don't get applied in July or August. So if you've got one of those early varieties and you get it off, don't go putting your post-harvest on in early to mid-August and think, oh, I'm done for the year, because that's actually going to be metabolized and utilized. You need to get post-Labor Day. So if you're putting on post-harvest sprays, you'll want to be applying that post-harvest spray post-Labor Day. We want to be getting into some cooler nights so that the tree isn't going to just simply metabolize. We want it to establish itself in the wood and the bud and in the roots, not to go to that year's growth. Sodium doesn't seem to be too much of a problem here. Next one, nitrate. We look at nitrate, we look at total N. I'm going to get back to that in a minute. Chlorine, sulfur, phosphorus. We'll come back to this in a minute. Here's a good one. We also look at iron, manganese, and zinc. These three pull and tug against each other. And what I mean by that is, think of this like a teeter-totter, a balance. And when one of these is really high, it puts downward pressure in the tree on those other nutrients. And the reason I'm really focusing on this one is this is a trend that I'm seeing a lot of. People are doing a great job of zinc in their trees. Now, whether it's because you're putting on your foliar zincs very diligently, which is a good thing, or whether you're applying pesticides that actually have zinc. Manzate, mancozeb have zinc in them. And we do pick it up quite substantially in the nutrient analysis with the sap that we're doing inside leaves. And I'm seeing, in a lot of cases, high zinc, but I'm definitely seeing a trend, early season leaves, those yellow chlorotic leaves you're seeing. If you're seeing them in your orchard, I have seen a trend here with iron and manganese being low. So it's not just iron, it's iron and manganese. And in not all, but a lot of those cases, I'm seeing really, really high zinc levels. In cherries, we're trying to get around 20 to 23 to 25 ppm of zinc in the early season, and by and go, I'm seeing 60s and 70 part per million of zinc right out of the gate early season. As soon as we got fully expanded leaves to go sample, and I'm finding those zinc levels through the roof. So you really need to start managing zinc levels, but really early season, if your leaves are pale, it might be iron and manganese. That's a trend I'm seeing here in Kelowna anyways. Boron, copper, moly. particularly boron in this case. Remember post-harvest, late season boron, because you're coming into bloom before you have a lot of leaves on cherries. So the real way to get boron ramped up to where you want it to be to maximize your fruit set is by putting it on last fall. It's also going to help with that bud for overwintering, by the way. So we want to be keeping an eye on that. But I am seeing a real mixed bag depending on where you're located as far as boron goes. I've got some growers that are very high on boron. I worked with Sarah last year on some post-harvest sprays, and we went and did some sap analysis in late August to get growers ready to know what to put in the tank for their post-harvest spray versus guessing. And we had a mixed bag depending where the growers were located. Some growers were so high on their boron levels, we didn't need to put boron into the post-harvest spray that normally would have been in there. Other growers were quite low in their boron, and we did need to put it in there. So just to say a blanket statement that your post-harvest spray should receive boron is a bit of a guess, and if you're wrong, it could actually hurt you. With a sap analysis, we are looking at silica. Silica is really neat. It is what we consider a non-essential plant nutrient, so the tree will grow fine without it, but it does play a role. It does end up in the cell wall structure with the calcium. Calcium gives you a thicker, thicker, stronger cell wall. The silica present in that cell wall gives you rigidity to the cell wall structure. We need to be careful because too much artificial silica applied to a tree will actually increase that rigidity to the point where the cells will become brittle. I've actually seen on other crops where too much silica causes splitting and cracking coming into harvest. So we don't want to over apply silica. We're thinking the ratio of silica to calcium, actually calcium to silica, I should say, is 5 to 1. So 5 parts calcium to 1 part silica. Looks like it may be the ideal ratio, but there's a lot of research still being done on that, and a lot of work still needs to be done. So if you are looking at a calcium silica foliar product, great. I would caution you 3-4 applications at most. And if you're starting to see some fruit cracking and your neighbors aren't, it might be due to the artificial calcium silica. One of the neat things about silica is that it is abundant in the Earth's crust. Just like granite, it's everywhere. The challenge is to get it into the tree, out of the ground and into the tree. For that to happen, it has to convert to an organic acid. That is driven through microbial activity. The soil microbes actually feed on the silica and convert it to an organic acid. And then that silicum, that organic acid, can now be absorbed by the roots of the tree and come in. And we actually see that in the sap test. And if we see high levels of silica, it can tell us that we've got a strong soil microbial population, and B, we've got healthy roots because it's being absorbed and it's coming in. If we have low silica levels, when we would expect microbial activity to be strong and roots to be functioning well, then we got to ask ourselves why. Do we got a soil health issue, or do we got a root health issue? And then we can dive a little bit deeper into what's going on in those trees. And of course, this ties in with the soil food web. I kind of slid this slide in the middle here just to remind everybody about soil health. When you're doing a lot of your pesticides and herbicides, there's an impact there. Don't forget that the anchor to those trees, the primary avenue to get a lot of nutrition into the trees, the root zone, and we do want to be paying attention to making sure we're supporting soil health. If you're seeing lots of earthworms around, that's a good sign. If you're not, that's a bad sign, and you need to be looking a little bit further into that. Let's talk for a few minutes about the nitrogen cycle. And I want to do that because I'm going to talk about nitrogen shortly. That's one of the things I do see over-applied on cherries. When we look in the soil, I think everybody's aware of the nitrogen cycle in the soil. If you were to put on urea, it's going to convert to ammonium, and then that's eventually going to convert to nitrate nitrogen. Well, there's a nitrogen cycle in the tree as well. If nitrogen gets taken up as ammonium, it will right away convert into amino acids, proteins, and fats, essentially be metabolized by the tree. But if the tree takes up nitrate nitrogen, and if you've got more of a neutral to basic soil pH, so if you're 6.5, 7, 7.5, 7.3 on your soil pHs, Most of the nitrogen in the ground is going to convert to nitrate readily, very fast, regardless of how you apply it, and it's going to come in as nitrate. Well, the tree has to convert that nitrate to ammonium inside the tree before it can utilize it and metabolize it. So we want to remember that. One of the neat things I like about the sap testing from NovaCrop is they break down those those types of nitrogen present in the sap in some detail. So we're looking at the amount of ammonium nitrogen inside the tree. We're looking at the level of nitrate nitrogen in the tree. And then we're also looking at the total nitrogen in the tree. And we want to make sure that those are in balance. Is the nitrate converting to ammonium? Typically, we want to see nitrate nitrogen in trees at under 20 ppm. If it's over 20 ppm, either you just put a huge application of calcium nitrate or something like that down and the tree hasn't metabolized it yet, or there's a problem. If there's high ammonium nitrogen, why isn't the tree metabolizing that? Or did you just do a huge application, the tree's busy metabolizing it? And remember, when the tree takes in nitrate nitrogen, so calcium nitrate is a prime example of nitrate N. When that nitrate N is coming in, it's bringing moisture in with it. So this time of the year, certainly calcium nitrate in can is a great product, calcium ammonium nitrate. But as you get closer to straw color and coming into more fruit color, you want to be avoiding nitrate N as much as possible, because when that nitrate N, it brings water into the fruit, and that's how we get a lot of cracking in our fruit. So we want to avoid late season nitrate N. When we look at the total N in the sap, we're looking at the nitrates, we're looking at ammonical, we're looking at urea, and we're also looking at amino acids and proteins. So to go back, when we look at this, that total N, that's everything, amino acids and proteins that are in the sap that have nitrogen as well. In a cold spring, when the tree isn't overly active, we're still getting nitrogen coming in. We're possibly, if there's a good balance with nutrition, getting it converted over to amino acids and proteins, but it's not being utilized by the tree yet. It's just kind of sitting there waiting. We'll actually see early season total N levels skyrocket. We're typically seeing, to begin with, we'd like to see ranges of the 300 to 500 ppm ranges. We'll see ranges as high as 13 or 1500 ppm. And it's not that you've over applied, we just have a lot of proteins and amino acids that have N in them, not going anywhere yet, not being utilized by the tree. Certainly seeing that this year in Kelowna, really saw it last year in Ontario. They had a really cold start last year in Ontario. Their ammonical was looking great, their nitrate was really low, their total N was spiking upwards of 2000 ppm. Once we had a week or two of warm weather, it was like somebody flipped the switch and we got that flush of new growth, but the total N came right back down to where we wanted it. So we're actually able to track that now with Sap Analysis, which is kind of neat. Molly's the forgotten nutrient. A lot of your soil tests that you're doing, if you're doing a proper detailed soil test, don't include moly. I'm kind of frustrated by that. I'll try not to get on my soapbox on this one too much this morning. Remember that conversion of nitrate to ammonical that happens inside the tree? Well, the enzyme that causes that reaction to take place so that the tree can convert nitrate to ammonical is driven by moly. It's moly dependent. Moly is an essential nutrient for plant growth. It's needed for that conversion of nitrate to ammonical. A&L don't regularly test for it unless you request it. If you request it, they'll gladly do it. It's not an expensive test. It's about $7 to add that to your to your detailed soil test. And when I asked them, I was actually in Ontario, swung by A&L Labs and had a sit down chat with them. I asked them why they weren't testing for moly, and their comment to me was nobody's asking, which I thought was kind of a weak answer. So I would encourage farmers that I'm working with, ask. You need to know where your moly level's at, because it can impact how your nitrogen is being utilized. So if you're getting soft fruit and you're not over applying the nitrate N, it could be because the tree can't metabolize that nitrate N and it's sitting in the tree as nitrate N because of a moly deficiency that you won't know unless you test for it. Chloride, let's touch base on chloride for a minute. Cherries are chloride sensitive, so are apples and a lot of other tree fruits. Doesn't mean you don't need chloride, it means you need very little. So putting on some chlorides at the right time is a good thing. Chloride acts a lot like potassium. It helps with the movement of sugars. It helps in the summer with the respiration and transpiration of the leaves for the tree. It's a good thing. It also plays a role in the metabolism of nitrogen. So a little bit of chloride is a good thing. Not a lot of people would put on a calcium chloride at the tail end of bloom. You're just going to fry leaves. But once you actually have some fruit there, once you're at that green BB stage, putting on some calcium chloride, if you're okay with the fact that it's relatively corrosive on your equipment, it really is an excellent source of calcium. And we still need to be putting calcium on at that time for maintenance to ensure that we got good calcium that's staying in the fruit. So where I like to tell farmers, put on your calcium chloride midway through the growing season, helps ensure that the nitrogen is being metabolized properly so you don't have a lot of residual N hanging around creating soft fruit. And it's a nice cheap form of calcium that works really well. I'm not going to get too much talking about reading the report at this time. That's really so much. When we're talking about antagonisms and synergies, this is breaking it down into a relatively simple perspective. Each quadrant tugs and pulls with each other. So when you look on the quadrant all the way on the left, you got calcium, sodium, potassium all together. Those nutrients would tug a war between each other for dominance. So we want to make sure they're well balanced. And then when you look across, there could be some antagonisms. Nutrient mobility within the plant. This is why we're sampling old leaves and young leaves separately. The tree is always trying to regulate the new growth first and foremost. And if it has an abundance of a nutrient it doesn't need right now, if that nutrient is mobile, it will move it into the lower leaves to store it. If it's not, it sits in the front and the top. And that's why when we're trying to determine, okay, what's that nutrient deficiency, we look at whether it's an older leaf or a younger leaf, because those mobile nutrients can come up and feed those younger leaves. So the deficiencies are first going to be seen in the older leaves. The less mobile nutrients can't come up from below when they're needed. So you see the deficiency right away in the new growth. So that's why when we're trying to determine, geez, is that magnesium? Is that sulfur? Is that iron? It really depends on where we're seeing it, old leaf, young leaf. But now when we take this knowledge and apply it to sap analysis, we can now actually see as the plant is building its reserves of some of these macronutrients, or it may be fine in the young leaf right now, but look, the old leaf, there's no more potassium. It's empty. The tank is empty. The tree's been feeding off of itself so long, you haven't experienced a deficiency yet, but you're on the cliff and you're about to go over. You better put something on to feed that tree. Here's an example of potassium and calcium. This is not the type of test result we want to see come back early season. High potassium, calcium is not moving into the younger leaves, not moving into the fruit. We need to get those potassium levels down, and there's certain things we can do to help with that. So there are some protocols to doing some sap analysis. I know that Molly offered to be involved in helping move some samples back and forth if there's some interest. We typically need to get a sap sample off, so the leaves need to be off the trees before 9 a.m. If it's a cloudy day like today, you have to about 9:30, 10 a.m. And that's for leaf tension and proper extraction. Also, if you think of that leaf, of what it is, it's a factory, and it makes nutrition all day and then digests it all night. So by coming in that early same window every day, we ensure that we're getting consistent results every time we're going out and sampling. So it's important from a number of aspects that the leaf comes off by 9 a.m. We are submitting old leaves and young leaves separately, as long as there's enough growth between them to get an old and young leaf off the new shoots. And we are shipping only leaves, no stems, just the leaves. The stem is the highway. We're looking at the sink called a leaf. And we do end up expedite shipping them. FedEx has worked the best for me. I do try to keep them cool when I'm sampling. I have a cooler in the truck when I'm sampling. I don't put any ice packs with them when I ship them, though. I just put them in a nice airtight shipping bag, and off they go. I shipped off some samples on Friday, last week, Friday. And I have the results. They landed on my desk at 6:30 this morning. So I'll be working with that grower today to go through them. So typically, if you ship them out Thursday, Friday, you're getting results Tuesday, Wednesday of the next week. So we're looking at a 5 to 7 day turnaround time. The trick is you got to get them to the airport and you got to get them shipped on FedEx with expedite. And that's where somebody like Molly, if you're traveling back and forth, might be able to help. talked a bit about this already. I've talked about the need for soil health. I think we're just about done, Molly. Soil testing. Sap testing, when you look at sap testing, I want you to look at it as an integrated science approach to nutrition. Sap testing is a great newer tool, but it's not the silver bullet. Growers need to be doing soil testing every 3 to 5 years, please. I've had some problems, and I went to a grower who I thought was a really good grower, and we're trying to figure it out. So he went and got a soil test for that block. And here we were, 2018, and the soil test was from 1994. Doesn't tell us a whole lot anymore. So soil test should be done every 3 to 5 years. When you combine that soil test to some leaf testing, to some sap analysis, to fruit analysis at harvest, particularly on apples, but even on cherries, but particularly on apples, you now have a really full picture to help you with your fertilizer efficiency. Fertilizer is not cheap. We want to make sure we're getting our best bang for our buck. So get the whole picture. There we go. Any questions for me, Molly?
Great. Thanks so much, Rick. That's excellent. I'll open the floor for questions and just see if there's any. You can text in your questions if you have any for Rick, or you can unmute yourself if you'd like to ask. And in the meantime, I'm going to just try to find that image, Rick, that I'd sent you that had come from a Creston grower, because I think that's kind of a neat case study sort of while we're talking about nutrition. So bear with me.
Rick, I have a comment I could make. And Rick, it was one of the things that you said that brought to my attention a study that we did a number of years ago in regard to the trace elements. You were mentioning the heavy use of zinc and the possible deficiencies of manganese and iron that may show up. And Mike Sanders was a horticulturist in Creston in the 1970s, and I was a summer student. And we did a survey in the cherry orchards in Creston. We sampled 120 trees to look at the nutrition of trees that had small fruit. And what showed up at that time was manganese toxicity, because orchards had been heavily fertilized with nitrogen, particularly 2100 prior to that, and it acidified the soil, and we had manganese toxicities that was creating small fruit. Mm-hmm. And it just reinforces the point in my mind that it's important for people to check the nutrient status of their trees, foliar, sap, and soil, so that they're not misdiagnosing things like little cherry virus for a nutrient problem. And it kind of connects our discussion this morning.
Very, very nicely done, Dwayne. Absolutely. Whatever tools you want to engage in, whether you want to take in the whole suite of tools, you don't guess, because those guesses are often incorrect. And it's just because so many different nutrient deficiencies can look like diseases or other nutrient deficiencies. I've spent 30 years doing nutrient work now, and I still need to send it off to the lab to make sure that I'm right. Some good comments. And yeah, you drop the soil pH, and of course manganese is suddenly coming in rapidly, right? And on tree fruit crops, we got to think of manganese, things like manganese and zinc. Bravo, ZN, well, the ZN and Bravo stands for zinc, and it's coming in.
Great, thanks both of you. There is a question from Dawn. What does NovaCrop charge per sample, Rick?
Oh, good question. The NovaCrop charges 22 euros per sample. So if you're looking at an old leaf and a young leaf, you're looking at 44 euros is the cost. Typically growers in the Kelowna area, what Grower Supply is charging growers in the Kelowna area, I believe it's $85 for a double set, which is an old leaf and young leaf combined. So yeah, about 85 bucks.
Great. And Rick had a question as well. When is the optimal time to do a soil sample?
Oh, that's a really good question. The same time you did it last time. I like to go out in the fall to do my soil sampling, but whether you sample in the fall or the spring, that can be accounted for in you know, what's just gone on. If we sample in the fall, we got to keep in mind that we just did a whole year worth of production on that soil, versus in the springtime that soil's had the winter to relax a little bit. But the key thing is choose the time that works best for you and your operation, and stay with it. Keep because that way we can compare samples. You also want to be choosing one lab and stick with that same lab. If you like A&L, they're a great lab. Don't bounce around between labs. Different labs have different extraction techniques and how they rate what that result looks like. So I can't take an iron, you know, a sample and compare the nutrients from the sample from this lab with the nutrient levels if that's that lab. So choose one lab, stay with them. And if you like sampling in the fall like I do, great. September, October is a pretty good time to go out there do a sample. But whatever time you choose, be consistent. Always go to the same lab and always sample at the same time of year.
Great, thanks, Rick. I'll show this image just quickly and let you make a couple comments on it before we wrap up here. So this was shared, I think you can probably see it now. Some images from this week from some of the Creston orchards. So we're seeing a fair bit of leaf yellowing that's showing up. And what do you think would be a good approach here, Rick, in terms of sampling this and taking a look at what could be done?
Well, again, it's, you know, I mean, just looking at it, when we see a lot of that, what I call intervenoclerosis. So the veins themselves are green, but the leaf tissue between the veins is yellow. That typically points to one of the photosynthetic nutrients, whether it's iron, magnesium, manganese. It's pointing to one of those. Again, if it's a young leaf, it's pointing more to the iron or manganese. If it's an older leaf, it's pointing more towards the magnesium, because magnesium is more mobile. So the younger leaf can actually pull it out of the old leaf, so you're going to see the deficiency. So those are some of the things to watch for. But certainly when that leaf is chlorotic like that, photosynthesis, that factory isn't optimized. It's not working at its peak. That means the tree's under a stress because it's not getting the food it needs. And that's the food it uses to ward off insects and disease because it wants to stay healthy. And that's also the food it's utilizing to grow a nice crop. So nice, heavy, sweet cherries, we need those leaves functioning. So when you see something like that, you need to be thinking the nutrients involved with that leaf function. And you want to be looking at those. In this case, like I said, I saw exactly those exact same color leaves in Kelowna 2 weeks ago. And we went and pulled a bunch of sap analysis. And 4 days later, lo and behold, low iron and low manganese. And I've seen that a few times in sap samples over last week as well, where we're seeing very low iron and very low manganese. So growers are going on at this early time of the year. I'm recommending them going on with some iron and some manganese and getting the results. If you don't want to do the testing, choose one of those nutrients, put on some iron. And if you're not getting a full response, maybe put on some manganese a week later. Iron is the type of product this time of year when you put it on foliar, and iron is really inhibited with cold, wet soils, by the way. If your soils are cold and wet, a lot of iron doesn't come in. Iron is one of those nutrients that trees respond very quickly to when you foliar apply it. So by putting on, if you suspect you've got a low iron and you put on a foliar iron, you would see results within 48 to 72 hours. If you saw some results and didn't quite match where you thought, then maybe you should look at some manganese as well. But certainly foliar iron, trees respond very quickly to it, and it's going to tell you what's going on. It's kind of a bit of trial and error, but it's an option.
Great. Thanks, Rick. So any final questions? I'll just put one last call out there for questions before we wrap up. Okay. Well, thank you very much, Rick, for joining us this morning. That was a valuable discussion, and thanks to Dwayne too for your discussion. Next week, we've invited Ken Sapsford from the ministry to talk about herbicide calibrations. And like we've said, we're certainly open to any suggestions on other topics growers would like to cover. So feel free to email Dwayne and I with any of that information. And have a great week, everyone. I should also mention our thanks to Investment Ag and to the BC Tree Innovation Fund for the funding towards this project. So we'll wrap up there. Thanks. Hope you'll join us again next week. Take care, everyone.
Molly.
Thanks, Rick. Appreciate it.
Take care, Dwayne. Take care, everyone. Stay safe.